huge amount of work done by a lot of people to enhance the UK Biobank resource. And now we're going to have a few presentations just about how the data from the first 150,000 genotyped individuals are starting to produce findings. So Jess Tyrrell from the University of Exeter are going to talk about understanding the causes and consequences of high BMI. Thanks, Jess.
Okay, thank you. So I'm part of a team of about 12 scientists at the University of Exeter who have been looking at the first release of the UK Biobank genetic data, and we got that data in June last year. And we've been doing a number of things, looking particularly at anthropometric traits and metabolic conditions. And we've been doing things like genome-wide association scans to try and identify some of those single nucleotide polymorphisms that were talked about earlier that are novel to particular traits. We've been doing some Mendelian randomization type analyses to look at causal relationships, and we've been performing gene environment studies. And today I'm going to talk a little bit about the latter two, so Mendelian randomization and gene environment interactions, to look at the causes and consequences of high BMI. So one thing we were interested in when we started with the data was to see if BMI actually caused differences in socioeconomic status. So there's lots of evidence in the literature that there's an inverse relationship between BMI and socioeconomic status. So higher BMI links to lower socioeconomic status. And it's generally thought that lower SES actually results in higher BMI, perhaps as a result of poorer lifestyles and diet choices. But we were interested to see if we could work out if actually BMI caused changes in socioeconomic status, and whether that could be due to something like discrimination, which may well have important policy implications. So we're effectively asking the question, if you had the CV of the same individual but sent photos of different weights, would that perhaps give a difference in the job as to whether you were hired or fired? So how can we do that? The standard observational epidemiology is really confounded. So what that means is there's lots of other factors that can be involved. And this is where we can use genetics to try and unpick the causal relationship. So if we take BMI genetic variants, so those are those single nucleotide polymorphisms that are robustly associated with BMI, we're effectively splitting our population into people that have got a lower BMI genetically or a higher BMI genetically, a bit like if you had a randomized control trial. And because your genetics are set at birth, we sort of randomize across all the various confounders. So we don't have to worry about confounding, and because we're starting with BMI genetic variants, we also don't have to worry about the sort of inverse relationship. So we can look to see how our BMI genetic variants are associated with SES. And that's what we did in the biobank. We created a BMI genetic risk score, and we looked at the relationship with 5 different measures. So things like income, deprivation index, and educational status. And what did we find? Well, when we looked observationally, we saw the relationship we were expecting. So higher BMI, lower socioeconomic status. And here we're looking at income. So a 1 standard deviation change in BMI was about a £1,260 reduction in annual income. When we looked genetically, the evidence wasn't quite so convincing. So although there did seem to be a causal relationship there, we had wide confidence intervals. So we decided to split the population into males and females to see if that made a difference. And what did we see? Well, in men, the observational association was much smaller, and we didn't see any genetic evidence of a causal relationship. But this is when it gets really interesting, when we look at women. So in women, there was a very strong observational association, and we also had robust evidence of a genetic causal relationship, suggesting that a 1 standard deviation increase in BMI equated to about a £3,000 reduction in annual household income. And we also saw a similar trend with the deprivation indices that we looked at. So this work was recently published in the BMJ, and we got some really entertaining headlines in the newspapers, one of my favourites being "Lardly worth it," which seems to suggest that there's some kind of disadvantage for overweight women. We didn't look into why that was, but it could be due to discrimination, things like lower self-esteem, or the fact that there are particular body types that are perhaps idealised, so cultivation theory tends to suggest more slender women are preferred. So I really want to highlight that Biobank is great for Mendelian randomization type studies. We've got a big sample size, which gives us good power. We've got lots and lots of genetic variants and a whole wealth of data within there, so we can look at a range of different predictors and outcomes. We can also do lots of different types of Mendelian randomization analyses, so we can try and account for various biases in our model. And obviously, with the 500,000 coming later this year, there is huge potential for some very exciting analyses. The second thing I wanted to touch on was whether genetic variants that are associated with obesity interact with specific aspects of the obesogenic environment or behaviours. So there's some evidence in the literature that suggests that genetic variants have bigger effects on BMI in obesogenic environments. So for example, if we took people who were physically inactive versus those that were physically active, there would be bigger effects in those physically inactive individuals. But a lot of those studies to date have got quite small numbers, they've had limited replication, and tend to rely on meta-analysis from multiple heterogeneous studies. And they've also not necessarily accounted for a range of statistical artefacts. And one of those that was particularly of interest is the fact that variation in BMI tends to be wider in fatter groups of individuals, which can lead to a statistical phenomenon known as heteroscedasticity. So why did we want to look at this? Well, if we can identify environments where we see interaction, then it can improve our knowledge of how best to target obesity. Is it the drinking of the fizzy drinks or the general sort of more sedentary time that's worse for individuals? So what did we do? Well, we took a range of obesogens in Biobank. Obviously, there's lots of variables we could go with, so we looked at inactivity, TV watching, dietary variables, and we split them into high and low risk groups, and then created BMI variables within those groups. And we created our BMI variables on both the residualized and an inverse normalized scale, which I'll explain a little bit more in a second. And then we looked at how those BMI variables associated with the genetic risk score. We calculated interaction effects using continuous measures, and then we tried to further account for heteroscedasticity using a number of statistical techniques, and performed a number of sensitivity analyses to check that the interactions were real. So what did we show? Well, on the left you can see we've got two sets of histograms, and what we've got is TV split into high and low TV watching. And on its normal scale, the BMI in our low TV watchers is around about 27, with a standard deviation of 4.5, whereas in the high TV watchers, it's 28.7, with a standard deviation of 5.2. We saw very robust evidence of interaction when the data was looked at on this scale, but then we changed it to an inverse normal scale, where you have a mean of 0 and a standard deviation of 1, so we haven't got that difference in variation. And whilst we still saw evidence for interaction, that was much weaker than we previously saw. And then we showed that we had evidence of interaction for a number of our variables, sedentary time, TV watching, and physical activity. So we then decided we wanted to look at whether these were real interactions. So we did a number of sensitivity analyses. We've simulated a continuous environment, which isn't associated with the real environment. So if we take physical activity, there's no association there, but it would associate with BMI in the same way as physical activity does. And we can then repeat that multiple times and look to see if the interaction effects are the same as what we see with the real variable. And also, because of the size of the UK Biobank, we can actually use a meta-heuristic approach to identify groups of individuals with identical BMI distributions. So if we take those who are physically active and those that are physically inactive, we can then effectively select individuals from across either of those two groups so that we can just look to see the effect of the BMI distribution on the interaction effect. So to conclude, we've seen some evidence of larger effects on BMI in obesogenic environments when we analyse BMI on the same scale, but we now need to do some further work to check if those interactions are actually real or statistical artefacts or non-specific. So just to very briefly conclude, I think the UK Biobank is a really exciting resource for gene environment and Mendelian randomisation studies. It's homogeneous, it's massive, we've got so many variables to consider, and there's huge future potential with the objective measures of physical activity and measures of biomarkers. And obviously, the 500,000 make it a very exciting prospect. Just to finish, that's a picture of the team in Exeter lined up in the order of one of their favourite traits. I'll leave you to guess what it is. And say thank you very much to the participants and people that work at the UK Biobank for making all this research possible.